All right, so we are ready back in the classroom to go ahead and analyze, look at our sample of leaf litter from the first site. This pillowcase was used to collect leaf litter from the site that was furthest from that really busy parking lot with lots of cars and foot traffic. As I said before, it does help if you label. I put a 200-foot label on the outside of this pillowcase just so you can keep track of what you got where. So I'm just gonna unknot this. You can use a bunch of different techniques for looking at your leaf litter. A lot of people find it helpful to use a piece of white poster paper to put down on the table, or even a tray if you happen to have a white tray. It helps to have something that is light colored so that the bugs that you're looking for can stand out. So I'm gonna undo this, and you just want to gently dump it out onto the tray. Try to get everything to come out. A lot of times, especially if you have pine needles, it'll stick to the inside. And I think we got most of it. And then there are different things you can use. Paint brushes, you can use little tweezers or forceps, but what I generally like to do is either have individual students or groups of students kind of just push through the leaf litter and separate it out against that white background and start looking for things. A lot of times they're very microscopic and not really obvious at first, but start looking for things that are moving. And there's a couple of different ways you can collect them. You can put them in a well plate like this, and you have the option to put them in the well plate and take photos of them with your cell phone, or you can also take photos with a microscope camera. Basically, this is a USB microscope camera. It's not actually what you would think of as a full large microscope, but we plug this into a laptop computer or a Chromebook, and you can actually put it on top of the bugs, take a photo, and if you don't really have time to analyze it and figure out what the name of that particular arthropod is at the time, you can look at it later and use that in your data collection. The other option, you can put some rubbing alcohol, not just in the well plates, but in these containers here, and that will preserve them long enough for you to go ahead and look at them whenever you have time to look at them if it's not immediate. So the first part of this, like I said, is just to kind of go through and start looking for things that might be considered an arthropod that you can include in your data.